My talk is titled Transposable Element Dynamics in Space and Time, and I will talk about two projects today. The first part is called Tirant stealthily invaded natural Drosophila melanogaster populations during the last century, and this is published work in molecular biology and evolution. And this will be the majority of the talk today. And then for the second part, I will present a project that I'm currently working on called the Unbiased Population Variation of Human Transposable Elements. So I would like to start with a bit of a general introduction to transposable elements. So what are these elements? They are selfish genetic elements that proliferate in host genomes, and we usually differentiate between different classes of them called the DNA transposons and RNA transposons or retrotransposons. And the main difference is the mechanism how they achieve an increase in copy number, with the RNA transposons having RNA intermediates while the DNA transposons do not. And between RNA transposons or retrotransposons, we differentiate between those that have a long terminal repeat called NTR and those that do not, called non-NTR. And these transposable elements, or TEs as I will refer to them for the majority of the talk, can make up a large part of genomes in eukaryotes. So in some plants, they can be up to 80% of the genome can be transposable elements. And in Drosophila melanogaster, we know that around 20% of the genome is made up by TEs, and in humans, it's much more, it's around 45%. And additionally to this abundance, there can also be a great variety in the number of families that can be active in an organism. So despite humans possessing many more transposable elements, there actually only are 3 or 4 known families that are active in human genomes, while in Drosophila, despite having a much lower total amount of Ts, there are many more different families that can be active, at least over 100. And what all these TEs have in common is that it's generally assumed that their activity in a host organism is detrimental, meaning that any host organism will try to repress the activity of a transposable element. And such repression can happen with various different mechanisms, including but not exclusive to different types of methylation signatures, specific proteins, including some crab zinc finger proteins that are important in mammals, for example, and various types of small RNAs. And the most prominent defense mechanism in Drosophila, which the first part of the talk focuses on, is called piRNAs or PV interacting RNAs. And these are a type of small RNAs that can prevent transposons from being active, for example, by an RNA interference-like mechanism that binds transcripts of Ts and prevents their translation. And these piRNAs are produced from specific source loci called piRNA clusters, which are genomic locations that are necessary to produce these piRNAs. But to successfully establish the piRNA silencing additional to these sequences present in a cluster, it is also necessary that some processed and built piRNAs are inherited from the mother to the offspring. So these piRNAs are actually inherited in the oocyte, which means they can only be transferred from the mother and not from the father. And this fact is very important in a specific phenotype called hybrid dysgenesis. So hybrid dysgenesis is something that has been studied a lot, and it's a very prominent example of how transposable element activity can directly have direct consequences on an organism's fitness. So it's phenotypes in Drosophila, or more specifically Drosophila melanogaster, that are induced by uncontrolled activity of a transposable element. And this activity is caused by the absence of maternally inherited piRNAs. So this means that if you conduct a cross of 2 different flies, where one has an active transposable element and an active host defense against it, and the other one does not have either of the two, where the one that does not have a defense is called the reactive strain, and the one that does have the active TE is called the inducer strain. And if you use a female that does not have the active TE, it cannot inherit the piRNAs. And what happens is that in the offspring, you will have activity of a transposable element. In Drosophila melanogaster, we know of 3 different transposons that can cause phenotypes due to such a crossing scheme. And these three are called the P element, the hobo element, and the I element. And what you can see here is actually the phenotypic effect that P element hybrid dysgenesis has on flies. So this is an ovary in an F1 offspring generation that developed normally. I hope you can see my cursor. And this is an ovary after uncontrolled P element activity, where you can see that there are basically no eggs left, and this is a sterile individual. So this is of course, a major detrimental phenotype, which is caused by solely by the activity of a transposable element. And these phenotypes are actually, and the elements underlying them are actually really relevant for understanding transposable element dynamics and biology in natural populations. Because so when these phenotypes were observed, researchers actually did not know what causes them. So the phenotypes were known before it was known that transposable elements actually are responsible for this. And then researchers were interested in determining how these, how is it possible that some strains that they had in the laboratory actually did have transposable elements and the respective defense, while others did not, which actually led to these phenotypes. And what was figured out is that these transposable elements, the P element, the I element, and hobo, actually had only just very recently arrived in Drosophila melanogaster. So frankly, they invaded the genome at some point after some of these laboratory strains that were used in the crosses were collected from the wild. So in this context, this means that invasions of transposable elements are very important. So I have a small cartoon here to illustrate how this usually works. So you have a specific species that has a naive genome that does not have a transposable element inserted. Transposable element is shown by this red hexagon, I guess. And then a transposable element enters the species. One of the example possibilities how this can happen is actually horizontal transfer. The transposable element that is present in species B in this example enters the species range of A by horizontal transfer or introgression. And then there's no defense present in the organism, which means that he can increase in copy number for a certain time until the organism starts silencing the activity of the transposon, in this case shown by these small RNAs. And then eventually, over time, what will happen is that if the TE is not active anymore, then it will just accumulate random mutations. And eventually, over time, it will lose its capabilities of transposition, which is why they're shown here in gray. So none of these elements are actually active anymore. And this is what happened, for example, with the P element. It was never in the species, and then it arrived in the species at some point in the 1960s, roughly. And so what you can see here is a plot showing the abundance on the y-axis in reads per million over the consensus sequence of the P element, so mapping next generation sequencing reads of genomic next generation sequencing reads over the range of the P element sequence. And the reactive strain just has no reads mapping. So there is no P element in the strain, while there is a variety of P elements in this inducer strain. But this is not always the case. So if you look at the same scenario, but instead the P element, you look at it for the I element, you can actually see that both strains, the one that has a no defense and the one that has the defense, both have copies of the I element present. So the genomic reads map. But what you can see is that there seems to be two different types in the inducer strain, where some look very similar to the reactive strain. So the colorful bars here actually represent SNPs compared to the consensus sequence. So what this means is that this is a very decayed, probably very old sequence that likely cannot produce any proteins anymore. While for the inducer strains, you have a bunch of gray area on top of here, which means those are rather new sequences that are likely still active. And additionally, it also has a couple of very old sequences. So what does this mean? This means that it is very likely that we have a second wave of invasion, which means that you had this I element, for example, being initially active in a population and reaching this state of all of the elements being not active anymore. And actually, this probably took on for so long that the defense against TEs has also decayed, because it was not anymore necessary to maintain, because no transposable element was able to be active anymore. And then eventually a copy from a different species arrives in the species range of species A, Melanogaster in this case. And since there is no active defense anymore, it can just proliferate again and produce new copies. And this is how you have the second wave signatures of new and old insertions. So this is something that we know has happened several times in the last 100 years, at least 3 times. And basically the motivation was to understand or to investigate if there are more examples of such recent T invasions, and if there are, to maybe find a reason why there is such an accumulation of these T invasions in Drosophila and Melanogaster in very recent years. So what we did in the beginning was that we basically compared 2 different laboratory strains. The one is called Canton S. It's a wild-type strain that was collected in the 1930s, so it's a very old strain that has been maintained in many laboratories since then. And the other strain that we looked at is ISO1, which is the strain where the Drosophila melanogaster reference genome is derived from. It's not a wild-type strain, but it's mainly derived from strains that were sampled much later than Canton S was. And what you can see here is a pairwise comparison of abundance. So everything on this side is more in ISO1 and everything on this side is more in Canton S. So we see a couple of different transposable elements that have strong abundance differences, for example, hobo, which is expected. The P element is not here because it's not present in either of the 2 strains. What you can see here, the elements tart B, tart A, and tare, those are telomeric elements, so they are partially domesticated in Drosophila, which means that these abundance differences likely do not really correspond to natural dynamics of Ts, because they're relevant for fitness of the host organisms. And there is one other transposable element which has a striking abundance difference between these 2 strains, and this T is called Tirant. And Tirant will be what we will be looking at this part of the talk mostly. So Tirant is an LTI retrotransposon, which is different to all the other 3 Ts that we talked about before. The P element and hobo both are DNA transposons, and the I element is a non-LTI retrotransposon. And Tirant is even, some people think that it has potential to be an endogenous retrovirus. It has a rather long sequence over 8 kb, and it's a member of the gypsy family of LTI retrotransposons. And it's not entirely clear if the activity patterns are restricted to soma or germline, respectively. And if you look at the abundance of Tirant in ISO1 and Canton S, similarly to the plot that I've shown previously for the P element and the I element, you can see that Canton S actually has some sequence remnants of Tirant. So you can see that there's some mapping. It's not like the I element where everything, the P element where everything was completely absent, but everything that we do see here has a lot of these colorful bars, so SNPs. While in ISO1 we have a lot of gray areas, so a lot of sequence, and it's also generally a lot more continuous. So it seems like the whole sequence is present similarly. So this would indicate that we have, again, 2 different types of insertions, old ones that are decayed and not active, and new ones that are close to the consensus sequence and active. So I will refer to these 2 ideas as consensus and degraded types of insertions, respectively. And you can see a very similar pattern if you look at genome assemblies for the 2 strains. So what you can see here on the x-axis are for the different chromosomes, just over the range of each chromosome arm. And the dots are annotations of Tirant. And the y-axis represents the divergence to the consensus sequence, meaning the higher up on the y-axis you are, the more different you are from the consensus sequence, the more SNPs you have, the more colorful you would be on the previous plot. And the size of each of these dots actually represents the size of the insertion. So the bigger the dot, the bigger the element. And what we can see in Canton S is that the only types of Tirant insertions that we have are very diverged, at least over 10%. and they are only found at the ends of chromosome arms, or mostly, which is generally believed to be heterochromatic regions. And yes, while in ISO1 we have the same types of elements diverged located in heterochromatin, but we also have a different type of Tirant insertion, which is mainly or often in euchromatin, much bigger and much less diverged. So again, this seems to be two different types of insertions, canonical and degraded. Where only, yes?
One question. In the Canton is chromosome 3R. There would be also an insertion like more or less close to the middle. This would be
Yes.
so that would be also a chromatin, right?
Presumably this would be, so these are, some of these annotations are a bit broken up because they are very diverged, so it's hard to infer. But it's possible that there would be one or two insertions of these old types in euchromatin as well. The general idea why they are more in heterochromatin than in euchromatin is just that there are selection constraints for stuff remaining in euchromatin for a long time. Therefore, the assumption here being, because they are very diverged, that these are much older elements, and therefore just by some type of selection constraints would be more likely removed from euchromatin compared to heterochromatin. Therefore,
Okay,
it's
thanks.
just they're more abundant in the heterochromatin.
Florian, another
Yes.
question. It seems that the X chromosome of Canton S is bigger than of ISO 1. Why?
Yes. That's just differences in the assembly. So we're using the reference genome here, but the reference genome actually in euchromatin is not the gold standard anymore. And this assembly that we did for Canton S was just longer in respect to the X chromosome.
Thanks.
Yes, so if there are no more questions, then yes. So as I said, we assume that there are these different two different types of elements, and as I just said after Sarah's questions, the assumption is that the canonical elements, the ones with the low divergence, are much younger than the ones found in heterochromatin. And to test this, we looked at population frequencies. So this is a sample of population. So it's a pool sec of different individuals of one population, sampled from France rather recently. And what we see here is, what we measured here is the frequency of the different insertions in the population. And the assumption is that older elements will have high population frequencies just due to a random chance, while everything that was active rather recently will only be present in the few individuals where the activity occurred and the direct ancestors, therefore they will be in low population frequency. And what we can see is that most of the elements that are located in heterochromatin are in high population frequency, which we have seen that are the ones that generally show high divergence values, while all the elements that are found in euchromatin have very low population frequencies. So those likely are the canonical elements with the low divergence values. And yes, so basically based on these results, we now formulated the hypothesis. Tirant recently invaded the Drosophila melanogaster genome, very similarly to the patterns that we see for the P element, the I element, and Hobo. And to more thoroughly test this hypothesis, we now wanted to look at sequencing data from a variety of different strains representing basically different points in time of wild-type populations. So we contacted SOX centers and looked for all of the different strains available in laboratories where we know for sure when they were sampled. So if you look at this plot, you can see here the year when this respective strain was sampled from the wild and established in the laboratory. So we were able to find strains sampled between the year 1925 and the youngest that we show here is 1967. And so we measured the abundance of Tirant as well as for the three TEs where we roughly know when and how they should have invaded the genome, the I element, Hobo, and the P element. And the color coding here means that red is the complete absence of an element, Yellow just means that you have degraded old fragments with a lot of SNPs that are likely not active, and green means that you have at least 1 or 2 sequences that seem to go over the whole species range with a rather low amount of SNPs, and therefore is likely a potentially active element, a consensus element. And what we can see is that strains sampled before or within 1938 do not have any I element, hobo, or P element, and usually also do not have any Tirant. There is the exception of the Urbana S strain, which has 1 or 2 different copies of Tirant that look a bit more complete and a bit more full length, which could be an indication that this is roughly the time where Tirant arrived in the population. The problem here is that it actually doesn't really make that much sense that the general copy number in the strain should be so much lower than everywhere else, because after the establishment in the laboratory, Tirant should still have been active within the laboratory population, similarly to how it was in wild types. So we're not entirely sure if this strain is reliable, but the important thing is that it seems that it wasn't present in most populations much before the time of 1938. While sadly there is a gap in fly samples here, but the samples that we do have again stem from the early 1950s, 1952 and 1954. And Florida 9, I will just say immediately, is likely just an artifact and was probably contaminated, because the presence of P elements in a strain collected from 52 is very unlikely, especially compared to everything we see afterwards. The P element is not suspected to have arrived in populations until like more than 10 years later. So this is probably not reliable. But either way, the strains that we do have that are more reliable from 1954 show now that Tirant is always present, and the I element is always present as well. While hobo is present in some of them, but not in all, and the P element is not present in any of them. And then if we sample strains from the 1960s, at some point the P element shows up as well, but always hobo, the I element, and Tirant are all present. So what this means is that likely for the recent T invasions of Drosophila melanogaster, that Tirant and the I element have probably shown up around similar times, something around the time frame of before or around 1938, until in the 1950s, they seem to be present in at least all of the strains that we were able to look at. The hobo element likely arrived a little bit later than the I element and Tirant, and the P element probably was the latest of the 4. Obviously, there could be a geographic variation coming into play as well, but this seems to be the most likely scenario from the data that we were able to amass and look at. And now we wanted to analyze, so we have now looked at population history. So the question now was, how does it look today? And it seems like Tirant is present in all populations that were sampled in the recent, so since 1967, any strain that was sampled, or even since before that, has consensus Tirant sequences. And if you look at principal component analysis using the single nucleotide polymorphisms of the different Tirant insertions, including not only the laboratory strains that I have shown on the graph, but also including several individuals from the Drosophila global diversity lines. So basically, these are fly populations sampled from 5 different continents: Beijing in Asia, Ithaca in the United States, so the Americas, Netherlands in Europe, Tasmania, which is close to Australia, an island, and Zimbabwe representing Africa. And you can see that the old laboratory strains that do not have active Tirant, the consensus Tirant cluster very differently from the other strains. But you can also see that, for example, the ISO1 strain seems to cluster a little bit distinctly, which I will come to in a second. And also, probably more importantly, that populations from Tasmania seem to have distinct SNP signatures. And we were a bit puzzled by this observation. One possible explanation that we have for this is that it could be some kind of bottleneck effect. When the Tirant invasion basically spread towards the island of Tasmania, that a specific element, a specific version of Tirant was spread there that carried a few SNPs. And actually, if you compare, so on the top here we see a mapping profile for a strain from Tasmania, and the red arrows indicate the presence or absence of SNPs that we see in all other populations. So, for example, these two SNPs here are not present in the other populations that were compared, or these SNPs in the LTR as well, while this SNP here is absent from Tasmania and present in the other one. So there's some differences that are specific to this region of Tasmania. And similarly, we also see some specific differences that are only present in ISO1, which is a non-wild type strain and the reference strain. So this was something even more puzzling and something that we never really found an explanation for. But it seems that there are some fixed or nearly fixed single nucleotide polymorphisms that actually are normal for Tirant and just very special in ISO1. But since ISO1 is the reference individual that was used to create the consensus sequence, those are shown as SNPs here, even though likely they shouldn't be in the consensus sequence, should probably be updated as ISO1 being the weird outlier here. One of the last things that we wanted to look at in terms of characterizing this invasion was to look at the piRNAs, so at the defense system active against Tirant. And what we see comparing a strain that does not have Tirant in Canton S, canonical Tirant, versus a strain that does in ISO1, is that there are no piRNAs defending against canonical Tirant in Canton S, but there is a ton of them in ISO1, which is what we would have expected to see. But if we look at piRNAs that map not to the consensus sequence, but to the degraded sequences that we have in the genome, we can actually see that there is a certain amount of piRNAs that are still produced that likely correspond to the old sequences, to the degraded insertions. And if we look at these 4 different strains, we can actually see that the abundance of canonical and degraded piRNAs is rather similar for everything that is not ISO1. So ISO1, again, is a weird outlier in terms of Tirant and seems like it should not be used as a gold standard. But so these are different populations from the GDL, from the global diversity lines. So B stands for Beijing, E for Ithaca, and so on. And so the basic idea is that the amount of piRNAs against degraded and canonical Tirant is very similar. But it is still rather, or at least in my opinion, it's still rather obvious that these degraded piRNAs are not capable of preventing the activity of novel Tirant. Because if they were, then since they are present in the strains that were wild type before the invasion should never have happened in the first place, because if these piRNAs could silence canonical Tirant, then they would have, and it wouldn't have spread through the populations a second time. And then we were...
A question. I do not really understand. Why would you have piRNAs against degraded elements? So, because I think most of the degraded elements are not sitting in piRNA clusters, so this is neutral divergence, and so I don't understand why they would be generating piRNAs.
I'm not 100% sure why they are generated either. I would imagine that some of them actually are still present in piRNA clusters, and this is the reason why. But I have never, I do not know for sure actually why they are still. I would imagine that it's just something that never was stopped. So they just were sitting in the piRNA clusters and are still somewhat produced from there. But
if an element is not active anymore, then you would find all the dead copies diverging independently from each other. And so I think there could be maybe one copy that could be whatever a degraded in the piRNA cluster, but then this shouldn't match all the others. You know, this.
Yeah. So what I actually did here is I used all the different copies. So this matches any degraded piRNA. It's not a specific consensus sequence. So.
Yeah, this is exactly what makes me so nervous about this, or so what I question is, I would think it could match a single one, but I'm very surprised that it matches across all of them. That's
Yeah, so it doesn't necessarily match across all of them, because if it matched to any of the different degraded insertions that we had, then I counted it as an abundant T. So this is more of a pool of piRNAs matching to different versions of degraded Tirant. If you know what I mean.
But maybe we can discuss later.
Sure. Yes, and just in the end, we wanted to check if there were some signatures for horizontal transfer. So for example, for the P element, we know that it was not introduced from a direct relative, but from a bit more distantly related species called Drosophila willistonii. But when we tried to make a phylogeny based on, so this is actually a phylogeny based on SNPs, and we did not find any SNP frequencies, and we did not find any difference from the phylogeny of Tirant and from the expected phylogeny of the Drosophila melanogaster species. So this, but what we did find is that there's a striking similarity of the consensus sequences of Drosophila similans, the pretty much closest relative, and Drosophila melanogaster. That is a bit more similar than would be expected by chance. So if there was a horizontal transfer event, it was likely from Drosophila similans, but based on the phylogeny and the other analysis we did not have any strong evidence suggesting this. So finally, I want to summarize the key findings from this first part of my talk. We have seen that Tirant has invaded the Drosophila melanogaster populations likely between 1938 and 1952. Canonical Tirant is present in all populations found today. And we have some that show distinct SNP signatures for one reason or another. Degraded piRNAs are likely not able to prevent, were not able to prevent the invasion and can likely not silence Tirant activity. And if there was any horizontal transfer event for the second wave of invasion, it would have been likely from Drosophila similans or another very closely related fly and not something more divergent. And open questions that one could tackle as a follow-up for this would, for example, be to ask why there is such a high frequency of recent transposable element invasions in the species. In the last 100 years, there were 4 different invasions, and it would be interesting to look at the reasons for why. And also to investigate in more detail if there is any hybrid dysgenesis produced by Tirant crosses. So we have been scanning for this a little bit and we have not found anything, but more thorough investigation would probably be needed to say for sure. And also something that I find particularly interesting is that this approach of having the snapshots, basically, so having laboratory strains from different points in times, this allowed us to reconstruct this novel invasion as well as the previously known invasions. And I was just wondering if there are possibilities to use available datasets or to start establishing datasets for other organisms to be able to do similar analysis. So I think that could be potentially interesting to think about. And now I will use my remaining time to hopefully quickly introduce you to the project that I'm currently working on, which is titled the Unbiased Population Variation of Human Transposable Elements. So this project is very much a work in progress. So I would be happy to hear any type of feedback you have, or also any types of ideas you have where we could take the project from the basic results that I will be showing today. And this is a collaboration with Ray Tobler and Yasin Suimi from the University of Adelaide in Australia. And so the underlying research question is basically, what is the extent of transposable element variation in natural human populations? And of course, this has been studied before, but pretty much all of the analyses that have been doing this so far have been based on the human reference genome. So a big focus of these studies was always to determine the respective insertion sites, so basically to know exactly where a transposon sits in the genome. This has, of course, a variety of informative uses. For example, for specific disease variants, it's very important to know the specific position of a transposable element. But just for quantifying the general abundance, it is not necessarily important to know where exactly in the genome a TE is sitting. And also, most of the studies that have been published focus nearly exclusively on 3 different TEs called L1, SVA, and ALU. for the simple reason that those are the ones that are believed to be currently or very recently active human populations, while all the other Ts are believed to have been silent for a much longer period of time. Therefore, most studies completely disregard many Ts and only focus on these 3. While in our study, we want to not do that. We want to look at all of the different transposable elements, and we want to do it in a way that does not involve a reference genome. And the reason why we do not want to involve a reference genome is actually, so there are several reasons, and I will try to explain 2 of them to you. So how do you find a transposable element based on the reference? Is that you have to use these bridging reads. So the idea is basically that transposable elements are repetitive sequences. So you have several times the exact same sequence in a genome. So when you use a short read sequencing approach, any of these reads could belong to any of these elements. And the mapping algorithm does not know where to put them. So it either assigns them randomly or does not assign them at all. The only way you can determine where in the genome a specific transposable element is sitting is by having these reads that are partially corresponding to a transposable element and partially corresponding to a unique region in the genome. Sorry, because then you can, for this light brown part, you can determine exactly where in the genome it belongs. And since part of it is a transposable element, then you would know, okay, this is where the genome, the regular genome ends in a way and the transposon starts. And on the other side, it's the same thing. So you know that this would be the end of this insertion. And but there are several problems with this approach. One of them being, for example, you can have, and this is something that happens quite a bit, especially in heterochromatic regions, you can have several transposable element insertions nested within each other. So in this example, we would have 5 different transposable element insertions all sitting within each other. But a reference-based approach can only identify a single one, because only on this very left and on this very right side, there is a genomic region that is unique, that could be assigned uniquely by a read, while everything in the middle here is the same. It would be unmapped or mapped randomly. And the number of TE insertions in this genome would be estimated to 4 less than are actually present. And another example is that you can have large amounts of structure variations in human populations. And this is actually quite important, for example, for African populations that carry a bulk of standing variation of all human populations. So African populations within themselves are more variable with structure variation than all other human populations combined. So if you want to be able to, and as I said, TEs can be very important also for clinical applications. So if you want to determine the presence or absence of TEs for clinical reasons, and you use the reference genome, and the individual has a large structure variation, which means that even TE sitting in a unique genomic region cannot be mapped to the reference because the whole unique genomic region is not present in the reference genome. So this, of course, is a major problem for not only for clinical studies, but also for generally inferring population variation. So how does our approach circumvent this? Actually, quite easily. It just ignores the reference genome. So the only thing we are using are genomic sequencing reads and consensus sequences. So yeah, you have to imagine for all human transposable elements, we roughly know their sequence, and there are available databases where you can see all the sequences for the transposable elements. And we can now take genomic reads and basically just map them to the consensus sequence. So in this case, all reads belonging to any of the orange insertions in the genome will be mapped to uniquely to this one consensus sequence. And so we could estimate just an average coverage based on these on the average amounts of reads mapped per base pair to the consensus sequence. But an additional thing that we do in our approach is that we establish a set of single copy genes. So these are genes that are always single copy and very conserved throughout all individuals, including, for example, the variable African individuals. And by estimating the average coverage of something that only occurs once in the genome, we are able to estimate the amount of copies a TE should roughly have in the genome. So if a coverage of, if a normalized coverage of 1 based on a single copy gene is compared to something that is twice as abundant, that would imply that a TE has 2 copies in the genome, 3 times as abundant, 3 copies, and so on. I hope this makes sense for everybody. So this is the basic idea. We do not need to use the reference genome. We do not need to look at where in a genome it sits. We just know there are so and so many TEs in population A, in population B, and so on. And this we can use to do population-based or individual-based comparisons. And for this reference sequence library that we use for mapping, we use the 965 different TEs available from RedBase, so they're publicly available, including these 3 active TEs, L1, SBA, and ALU. And for L1, it even includes several different variations that are known, that are like subfamilies. And additionally, we include over 400 single copy genes that we have determined ourselves. And they are present on all of the different chromosomes except for chromosome 21 and the Y chromosome. And to estimate the average copy number of 1, we only use autosomes, because the X chromosome obviously in males and females has a very different abundance. So any genes on there cannot be used for this, but they can still be used to basically as a double check, as a sanity check, if the sex assigned for the sample is actually correct. And the data that we are analyzing stems from the Human Genome Diversity Project, the HGDP, and it's Illumina sequencing of over 900 individuals from all different continents. So it's the regions are differentiated as Europe, Middle East, Africa, Central and South Asia, East Asia, Oceania, and America. And for each of these continents, we have different population samples. Yes, so now you have to imagine we basically just map all of these different sequencing data to our reference library containing TEs and single copy genes, and now we can estimate for each sample the average number of copies for all over 900 TEs. And this we can now okay, first a proof of principle. This is for a single sample, the average coverage for all single copy genes marked by a blue dot each, and all transposable elements marked by a green dot each. So you can see that all the single copy genes have a very similar coverage, indicating that they are all in very similar abundance of 1, while the TEs are a very different abundances. What was this? And yes, so most of the plots that I will be showing today are on a log scale, for the simple reason that there are certain transposable elements, most prominently ALU, that are just much more abundant than anything else in the genome. So ALU has several hundred thousands of copies. So showing a non-log transform plot usually just means you see ALU and then everything else in a little corner in the end. And yes, so this is now, so the first thing that we wanted to do with this data set is to compare the general abundance of transposable elements in males versus females, because we assumed that there will be a different TE sitting on the X chromosome compared to the Y chromosome. So what you see on this plot is the mean abundance for each transposable element over all samples that are male, and the mean abundance for each of the 900 transposable elements for all samples that are female, plotted against each other in a logarithmic scale. And everything that is, for example, this here is more abundant in males than on average, and everything that's around here is more abundant in females on average. And if you look at this on a non-log transformed scale, you can see that you don't see much, because as I said, you have ALU here and then everything else here in the corner. But what you can see is that if you lay a regression line into this, it is a bit lower than 1, which suggests that TEs on average are a bit more abundant in females compared to males. So most likely this just means that TEs, or many TEs, are more abundant on the X chromosome compared to the Y chromosome. And if you do a pairwise comparison, only looking at the 30 TEs with the highest differences in mean abundance between males and females, this trend seems to hold up as well, because out of the 30 TEs that I'm showing here, 25 are more abundant in females compared to males, and only 5 are more abundant in males compared to females. And yes, on the y-axis, you have the relative difference in abundance. So not the absolute one, because the absolute one, again, would just be ALU and then pretty much nothing. And what you can see here, for example, is that a specific variant of L1, of the active L1 transposon, is among these 30 most differentiated TEs in abundance, and it is known that L1 is very abundant on the X chromosome. So this actually makes sense that this is part of this list here. And yes, so this is the absolute number. So again, it's ALU, and then not so much else. So absolute is just dominated by these very abundant ALU elements. And now the question was basically, okay, we know that there's a difference between males and females, but is there actually differences between the populations, or are they all very similar? So this is just plotting all the different values. So basically the highest dot here is the maximum abundance value observed for TE in any of the samples, while the lowest is the minimum value. And these are the 30 TEs again with the highest relative difference of the minimum and the maximum value for males and females separately. Since we know that males and females have differences in abundance, we have to analyze them separately now. And I just quickly want to highlight that again, L1, the specific L1 variant seems to have a large variation in the populations, which is something that was previously known. And this is true for both males, which you can see here, and females, which you can see here. And a quick analysis that I did shows that most of the low abundant females that are down here are from African populations. So again, demonstrating that Africa has a large variety of transposable element abundance, which is something that you can also see in this plot. So this is a principal component analysis of the relative abundance of all transposable elements for all the different individuals, colored by their continent of origin. And what you can see is that Africa, shown here in red, actually differentiates pretty clearly from pretty much everything else for the female samples. And you can also see a nice gradient here for this principal component, that there is a lot of shared variation, but also some difference going from the Middle East towards East Asia, with Central South Asia somewhere in between. If you look at this for males, the pattern is much less clear. And the African populations are not nearly as differentiated, but there's a lot of more stuff going on, which could very well have something to do with the specific dispersion patterns of the Y chromosome. I'm seeing I'm kind of running out of time, so I will try to very quickly wrap up the talk. So the next steps that we will take in the analysis of these projects are to go more on a population scale, and not on the continent scale, but to compare the abundance differences between and even within the different sample populations. And we also want to include some additional samples. And something that I just very quickly want to show in the end is that something that I think it's very exciting, it's the inclusion of ancient DNA. So if we include a Denisovan and a Neanderthal ancient DNA sample into this principal component analysis of females, we can see that their abundance of TEs is very different on average than all the modern humans, which is not unexpected, I think, but I think that this could be quite a cool analysis if we can account for all the different biases that ancient DNA can potentially induce. And also, just as a very last plot, so this is the pairwise comparison of a Denisovan and a Neanderthal sample in regards of their abundance differences. And you can see that there are some clear differences, and some TEs are more abundant in Neanderthals and some are more abundant in Denisovans. So comparing this to what we see in natural populations today, I think could be quite interesting. And with this, I'm at the end of my talk. I want to thank my supervisor, Robert Kochler, my collaborators on the Tirant project, Philip and Kirsten, my collaborators on the human project, Ray and Yassine. I also want to thank Otno, Eli Lucas, and my additional PhD supervisors, Chi and Christian, and everybody at the Institute of Population Genetics and the Graduate School, and all of you for listening.